Now continue with the serological test. There is one more serological test which is heme absorption test, and this heme absorption test is also based on the heme agglutinin protein. We have already studied the heme agglutination test, which are based on the heme agglutinin protein, on the ability of the viruses which are containing the heme agglutinin protein. The heme absorption test will also be based on the same protein, which is heme agglutinin protein. The technique is just similar to the heme agglutination test, and similarly it will be also the heme absorption test used for the detection of certain viruses in the tissue culture. Similarly, the heme agglutination inhibition test, there will be also the heme absorption inhibition test too, which are based on the basis of the heme agglutinin. Its principle, why it is used and how it is produced, it is based on the erythrocytes' ability. The viruses producing heme agglutinin will absorb the erythrocytes to the surface of the cells in which the virus has grown. On the basis of this ability, you can just simply diagnose that which of the viruses which have the ability to absorb the erythrocytes. Just like you can see in the PPT, the viruses have been captured inside the with the surface of the cells in which the virus has grown. At the bottom there are the RBCs. However, at the not at the bottom, at the supernatant, there are the viruses which are just captured with the RBCs. Therefore, on the basis of the heme agglutinin, we can also use the heme absorption test and heme absorption inhibition test. The procedure is suspension of washed RBCs is added to tissue culture tubes. Of course, it is also based on the red blood cells. The RBCs, the washed RBCs is added to the tissue culture tubes. Presence of the heme absorbing virus results in clustering of the RBCs. Of course, because it is based on the heme absorbing erythrocytes and heme absorbing viruses. So if the heme absorbing virus is there, then the clustering of the RBCs, just like we have seen at the surface, it will be done with the heme absorbing RBCs. Rosettes around the infected cells of the culture can be observed microscopically. With the help of the microscope, you can easily see that the clustering of the RBCs happened at the surface. Here you can see the heme absorption and agglutination inhibition, how it happens, how it occurs. These are the red blood cells. First of all, there are two different kinds of the red blood cells which are being used here, and they are just mixed with the virus. We can take the example of the orthomyxovirus or paramyxovirus particles. So first of all, the red blood cells in the tube, and then they will be just mixed with the virus particles, viruses, and similarly the antiviral antibodies from the serum. If we have the serum and serum has the specific antibody for that virus, or maybe they have the heme agglutinin proteins of that virus, and plus these serum antibodies will be mixed with the virus. So if they have the heme agglutinin protein, then they will have the heme agglutination, and if they don't have that protein, then there will be the heme agglutination inhibition, and virus will be neutralized and heme agglutinin will be inhibited. So application useful. Similarly, with the heme agglutinin and heme agglutination, it is useful for the detection of viruses which are producing the heme agglutinin proteins.